So we're now going to begin Chapter 5, which we're going to look at the structure and the function of the plasma membrane. Here you can see just some of that information to review, where you remember that a plasma membrane isn't just another name for the cell membrane. So it's surrounding the entire cell, and it's made primarily of these materials called phospholipids, which, if you remember, have this phosphate head and then these lipid tails. And it's a bilayer because there are two layers of the phospholipids facing in this direction, where the lipids are on the interior and the phosphate heads are on the exterior of the cell, so outside, as well as on the interior of the cell facing the cytoplasm. The plasma membrane has important functions, and probably the main one is controlling and managing what is able to come in or out of a cell. So you can see here that it's more than just simply phospholipids. So we'll talk about some of these additions here as we go forward. But it manages that. It also defines the border of the cell, and it communicates both inside and outside the cell. So it's going to receive information, and it's going to communicate some of that information to the interior of the cell. And then, as you know, in complex organisms like us, these cells actually are very often in groups called tissues, so that they are cells that are the same type of cell that are working together. So the plasma membrane functions to adhere one cell to another. The classic way we look at the membrane is as a fluid MOSAIC model, which was proposed, as you can see here on the bottom, in 1972 by these two scientists. And what we mean here is that it's fluid because it has this ability to flex and to change in shape depending on what's going on around it. That these phospholipids that you see here are all lined up together, but they aren't really adhering to one another. They aren't like bound together, so that there's movement within this membrane. The other thing is it's a mosaic because it has lots of parts sort of embedded in those phospholipids, and we're going to see what those are. So this is a review of phospholipids. You can see here's the part of the phospholipid that likes water, is hydrophilic. And then we have the parts that don't like water, the hydrophobic tails, with these different fatty acids, some of which have the straight chain, meaning they're saturated, and some which have double bonds, there's a little kink here, which makes these unsaturated. They contain a glycerol, just like when we looked at triglycerides, we saw a glycerol as a part of this. Only two fatty acids, however, and this phosphate group at the top. And this is sometimes called amphipathic or amphiphilic. The individual phospholipids are that because one side of them likes water and one side of them doesn't. So it just sort of shows that dual nature. Because of that, when these phospholipids arrange themselves, they form this very particular structure. They form a bilayer. And what that does is it allows for the parts that like the watery environment, whether it's outside or the watery environment inside a cell, are going to face those directions. And then the parts that don't like water are going to be found on that interior. So here you can see that. We can see it again here when we're looking at it sort of in a flat view, where a piece of it has been taken, and we can see that not only, here's the mosaic piece, it's not only phospholipids, but all of these different types of proteins, carbohydrates, and lipids which are embedded in the membrane. Some are going to serve different functions. Some are going to serve for communication between the outside of the cell and the interior. Some are going to serve as channels to allow materials to pass through. Some will have some other communication functions within a cell. Some here, you can see cholesterol actually helps the cell to not freeze or to break apart when it gets too warm, so it provides some stability. And we're going to look at some of those in some detail. One of the things, though, are the things which we call integral proteins, and they integrate within and through that membrane. So the fact they're proteins should remind you that proteins first start off as this long chain of amino acids, and that those R groups on some of those amino acids are going to determine how this protein ultimately sort of folds and takes its shape. So if you look at these structures here, 1, 2, and 3, all of them are showing kind of more elaborate proteins that have folded, but some of them, all of them where you see the color in here, these are the parts that are showing the R groups that don't like water. So on this long primary structure, if this was a primary structure of amino acids, within that sequence would be a group of them that were destined to be inside where the lipids are, the ones that are hydrophobic. And so that sort of, and they end up there. And then the ones that like water, the ones that have R groups that are hydrophilic, are going to end up on the outside or on the interior of the cell. And that kind of determines that shape. So within the DNA, which is the code for everything and the code for proteins, there is a code to make these different types of integral proteins that actually get placed in the plasma membrane. Here's some pictures just to remind you of the different amino acids, and the blue boxes representing their R groups, and that for some, it would sort of tell you a little bit about where some of those amino acids, where the amino acids and where those R groups would be found on those integral proteins, whether it would be in the interior with the lipids or whether it be in the outside, where you would find beyond the phosphate groups, so out on the outside or the interior near the liquidy environments. So it's just helpful to see that. All right, so again, some other things which are embedded in this mosaic of a phospholipid bilayer are things other than proteins, or proteins with some modification. So one of them are these things called oligosaccharide. And these oligosaccharide carbohydrates are ones that here you can see anywhere that says glyco has to do with a part of a carbohydrate. And the word oligo means few. So the glycoprotein that you see over here, it actually is a communication molecule embedded in the membrane, has a protein, which you can kind of see there in blue, but then it has this little chain of carbohydrates. So that's the oligosaccharide, because it's a small number of them. Versus, remember what we looked at before, we looked at monosaccharides, disaccharides, and then we jumped up to polysaccharides. Well, this is the sort of the intermediate level. So we have some of those monomer of sugars here, but not a huge number like a polysaccharide. Same thing with lipids. We can have lipids too that have an attachment on them of these little oligosaccharides. Notice though on the picture, if you look carefully, you notice that the glycoprotein, you can see the protein kind of sticking up here, where on the glycolipid you can't, and that's because the lipid part is going to be snuggled into this part of the lipid bilayer. It'll be on the interior. Some proteins are important on the surface of cells for cell recognition. So when a cell sees another cell, and it can interact with it. So we have immune system cells that have these receptors on them which are basically glycoproteins, And what they do is they're going to recognize what is self versus what is non-self. Now, part of the problem when we get into some of the viruses that we're seeing, so this is an HIV or human immunodeficiency virus, is that there are some of these viruses which they kind of fool the immune system in that these glycoproteins on the normal cell surface on this immune cell look at this as being self, this HIV virus as having some component that is similar to our own. So it makes it a challenge for our immune system to fight these types of viruses when they contain certain proteins on their surfaces that are similar to our own. Membranes are fluid. That means that they can move, they're flexible, but they still stay together. They don't just fly apart. So fluidity is going to be affected by a number of things. One is what type of phospholipids are there. The ones which have saturated fats, remember how those were usually solid or more solid at room temperature, they can pack more tightly together and they're going to be more rigid. Some of the phospholipids, though, will have unsaturated fatty acids too, or a combination of that. Temperature can affect the membranes. So when things get colder, they compress. And so that is going to affect how fluid that membrane can be. And then a third component is cholesterol. So when we think of this cholesterol as a good guy, because what it is, it's a lipid that's embedded in the membrane that kind of keeps it stable, so it doesn't fall apart, it doesn't get too stiff when it gets cold. And that's a stabilizer. This little video that you have on OpenStax and you're reading it will show you a pretty nice little animation of that fluidity, or fluid nature of the membrane. The other thing that you notice is that the plasma membranes are asymmetric, meaning that what you see on the interior, close to where the inside of the cell would be, can be different than what you're going to see on the exterior. So that's the asymmetric part. So they give you some examples here, and that some of the proteins on the interior actually are anchoring some of these cytoskeletal fibers that exist, where some of the exterior proteins are going to be things which are going to bind to the extracellular, meaning outside the cell, the extracellular matrix. Glycoproteins also are able to bind to things which the cell actually wants to take in. Now this next section is going to look at transport and how actually things do come into or out of a cell. And so we're going to talk about the cell as membrane again as being selectively permeable. Now, permeable means something can pass through, but selectively permeable means not everything can pass through. So there are some criteria that are going to allow things to either freely pass through one way or the other, inside or out, or pass through under certain conditions. And then there are some things that cannot pass through that will be impermeable. So this is an important feature because it's going to be so much of the control of cells of what they are allowing in or out. There has to be a certain condition that's always sort of maintained between the outside of a cell and the inside of the cell. And we're going to talk a little bit about that. One thing that is maintained is an imbalance, not a balance, but an imbalance of certain ions that is always maintained by a cell, and we're going to look at how that works. And then other things which we're going to look at is how certain things can pass through, either passively, meaning they just pass through, depending on how concentrated they are on one side of the membrane or the other, or actively, meaning the cell can actually use energy to move materials. And that's the topic of our next video. So stay tuned.